Welcome back, biology, and today we're going to start talking about chapter 4, which is cell biology. This is an introduction. We're going to talk a lot about cellular structure, cellular function. We're going to talk about organelles. This picture over here on the left is of the nucleus and of what we call the endoplasmic reticulum. On the outside of it, you will be able to look at a picture like this and be able to tell what's going on by the end of this chapter. But let's jump right in and let's talk about. So we've kind of already posed the question, what are all living things made out of? And the answer is, of course, they're all made out of cells. And these cells are the smallest unit that can carry out all of life's processes. We discovered them in the late 17th century using a microscope, kind of like the one pictured here, that is using a mirror to help, and a lens to help kind of fract this light down. And then it's got this tube with lenses in it to help kind of see the specimen that's being looked at. Ours now are a lot more streamlined, a lot easier to use, but this is where we started. So our first kind of remarkable scientist talking about cells is Robert Hooke, and he is kind of credited with discovering cells. He was using a light microscope, kind of like the one pictured before, and he was examining a very, very thin slice of cork, and he saw what we call a great many little boxes. And all these structures over here is what he was looking at. This reminded him of rooms where monks lived. These are called cells. And then let's take a picture of those. So Hooke was looking at these cork cells, and these are some cells from Macedonia, an area in northern Greece, above northern Greece, where they have a monastery with cells. And you can see that these kind of arching structures, each one contained its own little monk, looks kind of like these cells that we see in those images of corks. Our second kind of remarkable guy is Anton von Leeuwenhoek, and he was the first one to observe living cells. So Robert Hooke's cells were all dead, but Leeuwenhoek was looking at actual living cells. The reason that he was able to see these is Leeuwenhoek's lenses were 10 times more accurate than Hooke's. He ground his own lenses. This kind of extra bit of technology that he had allowed him to be able to do a lot more science. And so in 1673, he observed these cells pictured here with the green stripes called Spirogyra. And he called organisms that he could see animalcules, or little animals. We now refer to them as protists. And most of what he was looking at were protists. Things like when we looked at our pond water experiment. All right, so the cell theory. There are 3 parts to the cell theory. And these images here were drawn by these first two, Matthias Schleiden and Theodor Schwann. And Matthias Schleiden's contribution to the cell theory is all plants are made of cells. Theodor Schwann's a year later said all animals are made of cells. And then after both of them, Rudolf Virchow kind of meshed everything together, and he said that every cell on the planet currently comes from other cells. So let's define the cell theory. The cell theory is this. Number 1, all living organisms are composed of one or more cells, and that cells are the basic unit of structure and function in an organism. Remember that we can have just unicellular objects, unicellular organisms, so having one cell is enough to be alive. And then the third thing is cells only come from the reproduction of cells. In your body, you don't get more of your cells just by assimilating and touching things in the environment. You have to grow more and reproduce more cells yourself. We can now talk about the cellular basis of life. We talked about these 7 characteristics earlier. The one that we're going to add is the number 8 there. So they're still all cells have organized parts. They obtain energy from surroundings. They perform chemical reactions. They change with time. They respond to their environment. They reproduce, and they maintain homeostasis. But the last part there is all living organisms share a common history. So they're all related somehow. These are some plant cells that you can see through this leaf here. Kind of cool, you got this water here. It's a very macro view. All right, cell diversity. not all cells look the same. Even inside of an organism, not all cells look the same. If a cell has a different shape, then it is probably going to have a different function. Some functions that cells in the human body have are protecting the body, sending electrical signals. Blood cells are shaped like discs. Some blood cells here that have these concave discs, so they're kind of meant to hold oxygen and carry it around to the tissues. Fat cells here, their nucleus over here is very small because most of their structure is filled up with fat so that they can hold all that energy. Smooth muscle cells have these big stripes on them so that when they contract, they can squish together and form muscle contractions. The next kind of topic we're going to look at is cell size. Cells have many different sizes. A human egg cell is really, really big. It's about the size of a period. And giraffe nerve cells can be up to 6 feet long. They go from the top of the neck all the way down to the body. But most cells are microscopic, right around this 100 micrometer level here. Organelles, those things inside of cells, are about 10 micrometers. And then we're going to talk a lot about these this chapter. But it's important to note that like bacteria are really, really small. They're really, really simple. They're about 1 micrometer. They're about 1/100th the size of a regular animal cell. Or this one's a plant cell here. So really, really small. The reason for cell shape and size is determined by surface area. And this is the amount of the cell that touches the outside. And then volume is the amount of stuff that's inside of the cell. The ratio of these, how much surface area to volume they have, kind of limits a cell size. So in this one over here where the nucleus is taking up the whole entire structure here, there's not a lot of room to get anything else in there, any nutrients or anything, so this cell is probably not going to be very viable. And then this one, there's so much area that this nucleus isn't going to be able to get the things that it needs from its surroundings. It's going to be hard to get things from one end of the cell to the other. So most cells kind of tend to this towards this happy medium here. So surface area to volume effects. Materials needed by the cell, such as oxygen and nutrients, need to get into these cells. And for having a big surface area here, you'd think this is good for the cell because it can get lots of oxygen, it can get lots of nutrients into the cell. However, it also needs to move waste out. So if you get too many waste starting to accumulate in here, it's much harder to get them out because you have to move them much further to get to the outside. So in a very large cell, the volume of the inside is too much for the cell to get all the nutrients it needs, and it's not able to get rid of all the waste. You can see the surface area of this one large cube here is 5,400 micrometers squared. And if we take these cells and we make them 1/3 of the size, but cubed, now all of a sudden, because of all these extra edges that are together, we now have a total surface area of 16,200 micrometers squared. So much bigger, 3 times bigger in the same amount of space, just because of how the cells are created in their size and their shape. All right, so now we're going to jump into some cellular anatomy, and all cells have 3 main regions. They have the nucleus, the cytoplasm, and the cell membrane. Unless you're a bacteria cell, and then you don't have a nucleus. Sorry, bacteria. All right, the plasma membrane. This is an image here of the plasma membrane, kind of simplified. It is a fragile, transparent barrier that contains all cell contents. We can see through the plasma membrane, so it is not super, super thick, but it does separate the outside of the cell and the inside of the cell, creating this compartmentalization. And this compartment here can have different proteins, it can have different sugar concentrations, salt concentrations, all of that stuff. We call the plasma membrane the skin of the cell. Right? You've probably heard that mitochondria is the powerhouse. We're going to kind of talk about the rest of them. The skin of the cell is the plasma membrane. We also call it the cell membrane. And the cell membrane is not a static thing. It plays a lot of a dynamic role in cellular processes. It's made of 2 lipid layers, the outside layer and the inside layer. There's also this yellow molecule here is cholesterol, And there's a lot of cholesterol in the cell membrane, especially in animals. This keeps the membrane flexible and fluid. So it has a hydroxyl group on the end, which gives it this polar region because it wants to have that polar head on the outside, and then this carbon rings on the inside are nonpolar, so they bond really well to these tails. Yeah. The important thing is that cholesterol is not a fat. It's called a steroid. Steroids are still kind of types of lipids, but it's just a little bit different. All right, the next part, the cytoplasm. This is the region of the cell that includes everything except the nucleus and the cell membrane. So it's kind of in between. It contains all the organelles, it contains fluid, and there's cytoskeleton there in the cytoplasm. Now, if we want to talk about just the fluid part of it, without any of these blue structures here, we're talking about the cytosol. This is just the fluid part of the cytoplasm. And the cytosol has molecules and small particles in it, and it's important to note that even though this is fluid-like, it's almost like jello consistency. It has so much dissolved in it that is like almost like a gel, like a hydrogel, versus just a regular fluid. At the nucleus, like we said, not all cells have a nucleus. Some have their DNA free-floating out here in the middle, and these are our prokaryotes. The nucleus is the headquarters or the brain of a cell. We also sometimes will refer to it as the control center. It's this kind of bluish structure here. It holds and contains all of the DNA, and that's its main job, is to store DNA safely and protect it. And DNA, this deoxyribonucleic acid, contains all the building instructions for whatever organism we're looking at. If it's a human, it contains all the information for a human. If it's a plant, it contains all the information for a plant, etc., and so on. So there are two types of cells, two main types, and the first is prokaryotes. And like I said, this guy doesn't have a nucleus. His DNA is just kind of out here in the middle. It doesn't have membrane-bound organelles. It has very simple organelles, things like ribosomes. The information that's just kind of loosely stored here, we call it the nucleoid, because it's nucleus-like, but not a nucleus. And then remember that the prokaryotes are split into two domains, which are bacteria and archaea. So both of those are prokaryotes. Bacteria, these are very, very similar to the first cellular organisms, and then archaea comes here, and the split between domain archaea and domain eukarya is what separates us from the prokaryotes. So they're more closely related to us over here in animals, but they're still very, very far removed. All right, eukaryotes. These are organisms made up of one or more cells. They have a nucleus, where these guys have that nucleoid. They have membrane-bound organelles. These are things like mitochondria is a membrane-bound organelle. The Golgi complex, endoplasmic reticulum, lysosomes. Organelles are these structures smaller than cells. Most or many of them are surrounded by a membrane, and these carry out the cellular processes. These organelles are not alive by themselves, but they all contribute to the cell being alive. Because they have these extra pieces that are doing these parts, making energy, breaking down wastes, eukaryotic cells can usually get bigger than prokaryotic cells. We're going to note real quick for cellular organization that you can get these colonial organisms here. These are collections of genetically identical cells that live together in a connected group. They're genetically connected or identical, but they have no relationship to each other. In fact, they don't work together at all. They're kind of just living together, and they benefit by living together, but they don't specialize. There's not one cell that's really good at getting oxygen and one cell that's really good at making energy. They kind of just do their own thing. Versus our unicellular things, which are just living off of free-floating. They're like living out here on their own. Colonial organisms will kind of grow together, but they haven't quite figured out how to work together. And because of this, colonial organisms are usually not truly multicellular. All right, so for some true multicellularity, this is organisms that have many types of cells, or many types of cells that are all doing different functions. So as organisms have evolved, their cells have become more and more specialized. Eventually, they are able to survive independently. Groups carrying out a specific function are called a tissue. So a whole bunch of fat cells would just be called adipose tissue. A whole bunch of muscle cells would be muscle tissue. Nerve cells, nervous tissues. We have bone tissue, more muscle tissue. Remember that tissues make up organs, organs make up organ systems, and then organ systems finally make up organisms. So it's this moving from one stage to the next. All of these cells come from this precursor cell, or a similar precursor cell, usually called stem cells. We'll talk about those more later. All right, for 4.3, we're going to talk about the fluid mosaic model and organelles. Most organelles themselves have a phospholipid bilayer, so they're having these inside and outside, where the hydrophilic head is attracted to water, and the hydrophilic head is attracted to water, and the hydrophobic head is not. It doesn't want to mix. They kind of do this interaction, these non-polar interactions in here. We call this the fluid mosaic model because there's a lot of things going on inside of the cell membrane. And this allows the cell to be both strong, but allows it to be able to, like with this cholesterol, not be so rigid that it can still slip past other cells. It can move around. It can kind of warp and flex. And this is all due to this kind of interactions of these different things inside of the cell membrane. All right, the first thing to mention is membrane proteins, things like this glycoprotein, things like this channel protein, these peripheral or integral proteins, globular proteins. There's lots of ways that we can put proteins into the cell membrane. And however these are arranged here, the jobs are still the same. If they have different structures, they have different jobs. So this channel protein is going to be really, really good at moving things in and out of the cell, where this glycoprotein here is probably going to be involved here, where it can receive and send signals by detaching or attaching things to this glycoprotein part here. Blood typing is something that you've probably heard of. It is a membrane protein. There are membranes on the outside of blood cells that kind of allow the body to register what they are. Different people have different blood cells. So if we match up the people who have the same type, they won't have any kind of reaction. Like I said, this channel protein helps transport things in and out. There's lots of different structures for these guys. We'll talk a lot more about them in the next chapter. Right, for the nucleus is conveniently broken also into three parts. The first is the nuclear membrane, the second is the nucleoli, and the third part is the chromatin. And the nuclear membrane is a double-bound membrane here where it's got two layers of phospholipids. We call it the nuclear membrane. And between the two membranes, there's a little bit of fluid. There are these holes you can see here, these kind of lighter spots. This is formed by some fusion of the membrane and to create what we call nuclear pores. And the nuclear pores have a specific kind of purpose. They make this membrane selectively permeable. Things are able to move in and out. Versus the cell membrane, these pores are much bigger. So things are able to move much more freely from the outside to the inside and from the inside to the outside. I like to think of the nucleus as a kind of library, with the DNA being a whole bunch of books. You have to be able to come in to find a book, to check it out, take it out to the cell, make whatever product you're wanting to, and then bring the book back. So inside of this nuclear membrane, we have a similar to the cytoplasm substance called nucleoplasm. It's found in the nucleus. It's kind of this plasmoid structure, nucleoplasm. All right, the second structure is this nucleolus or nucleoli in the middle here. They're these small, dark, round bodies inside the nucleus. They are where we assemble ribosomes, and ribosomes are incredibly important to the cell. They are the site where we actually have protein synthesis, where we're taking the code from DNA and turning it into product, which is protein. protein is going to be the product of our DNA. So nucleoli make ribosomes. All right, chromatin. When a cell is not dividing, its DNA combines with protein, and most of the time DNA is going to live attached to protein. So here's the molecular DNA here. It is going to attach to these round proteins, and this forms this kind of loose network of bumpy threads. As you can see, it kind of looks like a string of pearls here. This is called chromatin. When that cell needs to divide, it will condense this chromatin in these tightly wound coils, and then condense those coils into tightly wound structures called chromosomes. And these chromosomes are dense, these rod-like bodies. Normally, this is actually 2 chromosomes here together. We'll talk a lot more about chromosome behavior when we talk about cellular replication. But just think that chromosomes are just really, really tightly bundled packs. Think of it as like if you're going to Amazon ship all of these little books, you're going to wrap them up in bigger packages and make sure that they're all secured. That's what's happening here. All right, ribosomes. We kind of mentioned these guys. These are these tiny dark round bodies. They are the ones here, these little blue guys in this image. They are made up of proteins and specifically RNA. And of the RNA, the ribonucleic acid, they are made up of ribosomal RNA, a type of RNA. This is the actual site of protein synthesis. There's a lot of these guys. Some of them are floating in the cytoplasm, some attached to the nuclear membrane, some attached to the endoplasmic reticulums, which we're going to talk about, and some of them just float freely in the cell. The ribosome membrane combination is the rough endoplasmic reticulum. And the endoplasmic reticulums are these 2 types of endoplasmic reticulum here. They are the rough and the smooth. And they're this fluid-filled system of what we call cisterns. Think of them as like tubes or canals. They are going to coil and twist through the cytoplasm, and they are about half of the cell's membranes, is this endoplasmic reticulum, because of how anytime that you have these tightly wound here, they're increasing their surface area, so there's a lot more membrane here than it looks like. This provides a network for carrying substances. I like to think of it as like a factory assembly line. Mostly the thing that we're making is protein. That's why we have all these ribosomes. So the first endoplasmic reticulum we're going to talk about is the rough endoplasmic reticulum, or the RER. There's this one here. It has all these ribosomes attached to it. All the building materials for cellular membranes are formed inside of the RER or around it, and then they're going to move on eventually. So there's a lot of membranes being made. There's a lot of proteins being made. These ribosomes not only build them piece by piece, but they also fold the proteins into 3-dimensional shapes and then send them to areas of the cell to either get finished or to start doing their job. So the amount of rough endoplasmic reticulum a cell has indicates how much protein it produces. So it's abundant in cells that make and produce protein and send it to the rest of the body, like insulin. So the pancreas has a ton of rough endoplasmic reticulum, but it's not going to be found in high abundance in cells that don't make proteins. So fat cells or that adipose tissue is not going to have a lot of this rough endoplasmic reticulum. In fact, it has very little of it. The next endoplasmic reticulum is the smooth ER. This is a continuation of the rough ER. It goes out past it. It doesn't play any role in protein synthesis. It doesn't have any ribosomes, so that's kind of your sneak peek there. It functions in making cholesterol to keep that cellular membrane fluid. It will metabolize fats, and it will detoxify drugs. So same thing in cells that do a lot of these things, you'll find more smooth endoplasmic reticulum. So in the liver, which is our #1 detoxifier of drugs in the body, you're going to find a lot of smooth endoplasmic reticulum so that it can detoxify those drugs more efficiently. All right, next up is the Golgi apparatus. You can refer to it as just the Golgi if you want to. Fun fact about the Golgi apparatus, it is named such because at this point in time in human history, when we discovered the Golgi, we were also doing a lot of factory working. So a lot of the cell components have these kind of names that feel like they're like from a factory, and that's because of the timing of this. So these are a bunch of flattened membranes here, and they produce these swarms of tiny vesicles, which are these small lipid capsules that have a double layer that are used to store things. The Golgi is usually found close to the nutrients. It modifies and packages proteins. Sorry, that got cut off there. Protein. Kind of think of these guys as the traffic director of the cell. They're taking things on one side, these incoming ones, and then they're exporting things on the other side out to the cell. All right, about these vesicles. Some of them are called secretory vesicles. Secretory means to secrete or to release. So we take these synthesized proteins, and they travel up to the plasma membrane. So this guy's moving up here. As it starts to reach the plasma membrane, this is going to fuse together, and it creates this kind of pocket that dumps out to the outside of the cell. So this can be used to get rid of wastes, to put proteins into the bloodstream. Like when you're getting adrenaline in your bloodstream, this is what's kind of the process that's happening. In addition to these secretory vesicles, the Golgi also makes lysosomes. And this is a lysosome. The name lysosome means breakdown body. And they come in different sizes, from really, really small to really, really big, depending on where they're from. They are these just membrane bags, and they have lots of these enzymes, these kind of orange things, inside of them. And these guys are like the demo workers of the cell. They're going to take in things that don't belong in the body or need to be broken down and use these enzymes to chew them up and get the important beneficial nutrients out and get rid of the wastes. So these guys are really common in white blood cells. White blood cells are kind of your body's defense system. They're going to go eat bacteria, eat them up with these enzymes, and then spit them out. So if for some reason a cell is oxygen deprived, let's say there's a wound or something, these lysosomes will rupture, and they will eat damaged cells or cells that have died. So these guys are really important in what we call programmed cell death. Once again, a lot of these names come from the factory era. Peroxisomes. These are also membranous sacs. They also contain enzymes, but how they form and where they form from is a little bit different. These guys aren't formed from the Golgi apparatus. So they're using molecular oxygen in this crystalline core here to detoxify harmful poisons and substances, things like alcohol and things like formaldehyde. They also disarm free radicals. And free radicals are these highly reactive chemicals with unpaired electrons. The reason that we don't like free radicals, the reason we try to prevent them, is free radicals can scramble your protein and scramble your nucleic acid structure, so your DNA and RNA. And we kind of like to have our DNA and RNA unscrambled. And some more things on free radicals here. Free radicals are the normal byproducts of cellular processes. We don't allow them to accumulate. If they do, the cell starts to die, starts to shrivel up. So peroxisomes will convert these free radicals to hydrogen peroxygen, H2O2. And it does this using an enzyme called catalase. Catalase is an enzyme that takes this hydrogen peroxide and makes it water, H2O. And then the important thing to notice here is even though they look similar to lysosomes, they aren't made by the Golgi apparatus, so they have a little bit different job. Next up is probably everyone's, the one that most people know, everyone's favorite, the mitochondria is the powerhouse of the cell. The mitochondria has a double membrane as well, separating the inside from the outside. Anytime you see this, it's because the organelle or structure wants to have a different environment on the inside versus the outside. So on the outside is smooth, and that offers protection so that the mitochondria can float around the cell and bump into things. And on the inside, it has these shelves called cristae. And anytime you see these projections like this, like these finger-like projections, this is to increase the surface area on the inside here. The reason we want this is the enzymes that carry out the reactions here to break down foods and produce energy need to have lots of extra absorption to do it. So they'll break down food to produce ATP. Something important to note is the mitochondria DNA. It has its own DNA in this ring, and you actually inherit your mother's mitochondria DNA. So it's way easier to track maternal lines because your mom will give you her mitochondria on the egg cell because there's no mitochondria in sperm. So everyone has inherited their mitochondria from their mother. Kind of a fun fact there. All right, adenosine triphosphate. We've talked about this molecule before. This is the ATP. This is the one that provides all the energy for cellular work. Liver and muscle cells have a high number of mitochondria. They do a lot of energy production. Inactive cells like fat cells don't have a lot of mitochondria. They're not as involved in making or breaking apart energy. They're more involved in just storing it. All right, next up, not really an organelle, but we're going to talk about the cytoskeleton. This is a network of protein structures in the cytoplasm, and they're going to act on the cell as a support system. They're going to determine cellular shape, support other organelles, and they're going to provide machinery needed to move things intracellularly. And going from largest to smallest, we have microtubules, intermediate filaments, and then the smallest one, microfilaments. And we're going to start on the smallest one, microfilaments. So microfilaments are things like actin and myosin. They're involved in cell motility. These kind of green ones here. They also help provide cell shape changes. So if a cell needs to go from a big cell to a small cell, or it needs to stretch out, these are the ones that are going to do that. Because of their contractile nature, these are very common in muscle cells, and they're what causes your muscles to contract. When you flex, you are flexing your microfilaments. Just a lot of them. The next is the microtubules. This green on this picture here is microtubules. This helps determine the overall shape of the cell. They also help do the distribution of organelles, and they're very important for cellular division. When we talk about cell division, where these guys will come back into play. The centrioles. So the centrioles are this really small piece on this image here, but blown up, they look like this. They lie close to the nucleus. They're found in pairs, and they're always going to be in these right angles to each other. They are made up of microtubules in there. And these guys are going to be very important when we talk about cellular division as well. So these guys will come back again. Cilia, these are whip or hair-like extensions that move substances along the cell surface. In the respiratory system, that's what's going to move mucus from the bottom inside of your lungs up the throat so that you can cough it up and get rid of it. These are formed from centrioles. So these guys have little centriole bases there. But these guys will undulate and kind of move things along, kind of like brushing things out of your body. All right, flagella. They're kind of related to cilia, but when they are longer, they're called flagella. In the human body, there's only one flagellum, and that is the sperm. This guy here is a paramecium, a protist. They have lots of flagella. Bacteria will have flagella. Yeah, the sperm has a single flagella called a tail. This helps to propel the cell. And flagella will be used for movement, like moving the cell through things. The cilia is used to move things along them. So they're not really going to, some of them will crawl with their cilia, but most time the cilia is used to just move things along. All right, next, we're going to talk about plant cells. And plant cells have most of the same organelles that we've talked about so far, but they have three differences. And the first is a cell wall, a large central vacuole, and then these things called plastids. All right, first up, the cell wall. This is this rigid layer outside of the cell's plasma membrane. It is made of a polysaccharide called cellulose, and it forms a stiff box around each cell. Cell walls have big pores in them, the same way that the nucleus does, to allow water, ions, and molecules to enter and exit the cell. Well, these guys still have the cell membrane underneath of this, so they still are selectively permeable, even with these large holes in the cell wall. These guys are still maintaining a nice internal environment. There are two types of cell walls. There's primary versus secondary cell walls. Primary cell walls are formed by enzymes directly on the plasma membrane, so they're right on this plasma membrane here, and they're only going to grow in one direction. The secondary cell walls, happens when this stops growing, and it grows on the outside of the plasma membrane. And it is very strong, and once these guys kind of solidify and are rigid, it can no longer expand. The reason for these cell walls and secondary cell walls are really important in the Pacific Northwest here, is they allow trees and other plants to be rigid and to stand up really tall. The central vacuole, this is this large fluid-filled organelles that storms water, and usually dissolved in that water there are things like enzymes, metabolic waste, or other materials. How these guys form is there's lots of small vacuoles that form like little tiny water droplets, and then when those, just like water droplets on the window, when they bump into each other, they merge together to form these really big vacuoles. Vacuoles can be up to 90% of the cell's volume, and they're going to push all other organelles against the cell wall, kind of like we saw in that fat tissue. All right, the effect of water on cellular structure. When water fills the vacuole, the cells expand and the plant stands upright, and it's happy and it's nice. When it loses water, the vacuole now has shrunk down to much smaller. These cells shrink and the plant wilts because it doesn't have that water anymore holding that structure up. Think of it as like a water balloon pushing on all sides, making that structure really tight so that that plant can stand up. So other vacuoles can be used to store toxic materials. The acacia tree here uses a vacuole to store poisons that protect them from predators. They also store color pigments. Lots of different uses of vacuoles in plants. Those are just a couple. The next are plastids. These are organelles like mitochondria in the fact that they have a double membrane and contain their own DNA, but how they make energy is a little bit different than the mitochondria. And there are several different types of them. The most common is the chloroplast. And the chloroplast uses light energy to make carbohydrates from carbon dioxide and water. Inside of the chloroplast, we've got these thylakoids, which one thylakoid here is a flattened membranous sac. They come in big stacks. They have a green pigment called chlorophyll. And chlorophyll captures and absorbs sunlight, except for the green color. It reflects the green color. So this is found in plants and eukaryotic algae, things like seaweed. They will have lots of chloroplasts in them. Chloroplasts just don't belong to plants. They can be found in other things. All right, chloroplast DNA. This is the chloroplast DNA here. It is very close to that of photosynthetic bacteria, specifically cyanobacteria. Chloroplasts can only be formed from other chloroplasts, and this evidence kind of points to the fact that we think that cyanobacteria are descendants of ancient prokaryotic cells that were kind of eaten up by another cell and not quite digested. And we call this theory endosymbiosis. So in endosymbiosis, we have an ancient prokaryotic cell getting incorporated into other ones. These cells then entered into a symbiotic relationship where both of them are getting something that they want. First, we think that an aerobic prokaryote was consumed. This turned into the mitochondria. And then later in the history, a cyanobacteria was consumed, which formed a chloroplast. This is why all plants have chloroplasts and mitochondria, and why we have mitochondria but not chloroplasts. All right, chromoplasts. This is a picture here of a carrot cell. Chromoplasts are very colorful plastids. They contain a lot of pigments that may or may not be used in photosynthesis. Carrot cells contain this carotene, which is an orange pigment that gives carrots their color. In carrot cells, are these cells providing photosynthesis? Is the orange part of the carrot involved in photosynthesis? No, it's the one that's under the ground. So this part of the carrot is not really involved in photosynthesis. He's under the ground. He's not getting a lot of sunlight coming down there into those cells. So not providing a lot of photosynthesis. And then some other plastids. These all share some general features of chloroplasts. They differ in their content, what they make. They can be amylioplasts, which store starch, chromoplasts for color. They can be leucoplasts. They can be all kinds of these. This one contains chlorine. But they all come from this common precursor called a proplastid. And this is what this guy is up here. All of these guys originated from here. So that is the end, the conclusion of chapter 4. I hope you have a much better understanding of cell function, cell structure, especially regarding to organelles. I hope you can tell the difference between a prokaryotic cell and a eukaryotic cell, and then in the eukaryotic cells, the difference between an animal cell and a plant cell. You guys are going to get some practice with this. Thank you for watching, and I'll see you in the next one.